So hello everyone, we are going to discuss about the isolation and purification of plasmid DNA today. So plasmid DNA isolation and purification involve extracting and purifying plasmid DNA from the bacterial cell, separating it from the genomic DNA, protein and other cellular debris to obtain a highly concentrated and pure form of plasmid for further analysis. So firstly we are going to harvesting and lysing. So harvesting bacteria containing the plasmid are grown and harvested through centrifugation. Resuspension. The bacterial pellet is resuspended in a buffer to prepare for lysis. Then we are going to lysis. The bacterial cell are lysed, broken down using a lysis buffer containing sodium dodecyde sulfate (SGS) and sodium hydroperoxide (NaOH). This disrupts the cell structure and denature both chromosomal and plasmid DNA. Then clarification. The lysate is clarified by the centrifugation, filtration, or magnetic clearing to remove cell debris and other insoluble material. So the separation and purification. The neutralization. First step is neutralization. The lysate is neutralized with potassium acetate using allow only the covalently close plasmid DNA to remove renal and remain solubilized. Then precipitation. The most of the chromosomal DNA and protein precipitate as a complex with potassium and sodium dodecyde sulfate, which is then removable by the centrifugation. Then further is binding, washing and elution. The plasmid DNA is then bound to a solid matrix जैसे कि silica column and washed to remove any remaining contaminants and finally elute in a suitable buffer. So this is the optional step. If using a silica column, the plasmid DNA is bound to a column, washed to remove contaminant and then elute into a suitable buffer. Final purification and storage. Purified plasmid DNA can be further purified using technique like ethanol precipitation or phenol chlorophyll extraction. Then storage. The purified plasmid DNA is then stored in the suitable buffer as -20 degrees Celsius or -180 degrees Celsius. Then here is the flow chart we are going to discuss. The cell are lysed using detergent, then disrupt the plasmid membrane. Then cell content treated with the protease and RNase to destroy protein and RNA. After centrifugation, cell debris pellet are bottom and the DNA at the supernatant. The supernatant containing DNA is transferred to a clean tube and precipitated and washed with the ethanol. Then using matrices of silica method, the DNA is bound to silica membrane spin column in the presence of high concentration at the catrophic salt. Then the contaminant are washed away. DNA is elute from the silica membrane in water or low salt buffer. So here is the about the plasmid DNA. Hope you can understand it well and hope you like it.